While I'm waiting to get online, it is 12:40, so I'm gonna start. So you've all now seen the email which says, we're not coming back after break, at least not immediately. The school will be closed until April 5th, that's my understanding, and all classes will transition online. I don't know anything about online, but don't you worry about that. I will deliver content. My plan is, I have a software program that will allow me to capture my computer screen and audio. And so I can lecture with slides and annotate them just like I do. It's just you won't be able to smell me. You'll be able to hear me and not see me. But hey, that might be a bonus for you, I don't know. So look for that. And I'll have to do that during spring break, I guess, get those lectures ready. So there will be lectures online, I'm certain of that. What happens to the lab? I have no idea what the answer to that is. I mean, we have one more week of lab experiments to do. So I'll maybe try to update you on Friday about that. We still, let's see, on Friday we have our mid-exam, so that's gonna go off without a hitch, I think. And then next week is spring break, and then we're scheduled to miss 2 additional weeks after that, which will mean 2 quizzes, which I'll have to administer online. So you'll still get those. I don't know how to do quizzes. So it might just be like a take-home thing, but maybe I'll get really insightful and figure out how to do a timed online quiz. I don't know.
You can do that
They
online.
have them.
Yeah, they have time quiz on that.
Blackboard.
I don't know how to do those.
Oh, that's one.
You have spring break, huh?
I'm old. I don't know if I can learn.
That's one.
And then there's a lab quiz. So let's just push all the lab stuff off till Friday. I don't know what's going on there. Just don't. But the lecture portion will be online. There will be quizzes online for those portions. And you'll want to pay attention during this period of time, because if I need to contact you, it'll be by email, and it'll be by the CCSU email. So you should monitor that. I would say once a day is what you should be doing. That's what I do. You should look at your emails once a day.
So the study guide questions on Blackboard, do those apply to the mid-exam? Like, you recommend that we should study them
I
for the
didn't post any
mid-exam?
questions from last lecture, but otherwise it's up to date.
Okay.
And I will tell you that when I go to make an exam, it's so easy for me to just grab questions from a study guide.
Gotcha.
And really they're there to help you. So here's what I will say. I won't guarantee that I'm just picking questions from there, though there's a bunch of them now, so it seems doable. But if you could answer those questions, you're ready for my test. And you will do well. Okay? I might have said draw the structure of serine, but I might not say draw the structure of serine because we're supposed to know so many other amino acids, and I might choose one of those other ones that seems reasonable. Anyway, you get the idea. I'll update today's lecture, and that will be included in the study guide, and therefore also include as material for the exam. Remember, the exam will contain laboratory type stuff, questions too. Good tea.
I have a question. I saw an email today about the school being online on spring break. So will this just be like our notes for class?
Will it just be what?
Will like the slides be our only notes for like a lecture when we're out of campus?
No. My plan is that I will record lectures post-break, so they'll be available to you by at least one lecture by Monday of class. So I'll make sure I stay current with the class schedule. So I don't know that I'll post all 9 or 6 lectures all at once, but I'll certainly have one ready for the Monday when you were supposed to come back, and there'll be one ready for the Wednesday and for the Friday, and those will be in the form of a video of some sort online. It'll be the slides, I'll annotate them just like I do, and I will speak into some sort of microphone, and you will have that audio and video as your class. That will be the online instruction.
The figure that's due next week, you want that on Blackboard?
Well,
now?
there's, right, so normally, if classes were going along normally, you would not owe it to me next week, it would be the following week.
Okay.
Just because nobody's here to get it, so you're not here to give it to me.
Oh, yeah,
So,
sorry.
so we will plan on that figure being due in my mailbox, that is my email mailbox, at your regular scheduled laboratory time. So if you're Tuesday morning, 8 a.m., I would expect it by 8 a.m. on Tuesday, not next week, but the week after. And if you're the 10:50 class, you would get it to me by 10:50. So no changes to that, yeah?
What are we doing about the poster?
We are doing a poster.
Are we doing that after we come back?
Well, so it's
Or
interesting,
do you
let's
want
see.
us to still be working on it with our group online?
I'm gonna go from memory about dates. But my understanding is next week's spring break, the week after that, we were gonna do the third laboratory. The week after that was gonna be advising, Tuesday. And then the week after that was gonna be the poster. That week after that, that makes it April 7th, which we're supposed to be back for. So it's possible that we'll still be having a poster session. Okay, but our data is gonna be somewhat limited, because I'm not clear how we're gonna get the third week's data or not here. But that, then I would group the poster together with the laboratory stuff and push it till Friday, and I'll have more clarity. Could be that I take your membranes and probe them with antibody, because I'm allowed to come in, and I can then post the Western blot for you. It's not optimal, doesn't give you any experience, but it does get you the result that you would need for a poster. And then the exam is April 7th anyway, 6th, sorry, 5th is a Sunday. April 6th is the scheduled exam, that's your first day back, according to the current schedule. But that might change too. And if it changes, we'll make changes with it. The plan is to still give you content and to still look for some sort of assessment factor. Assessment of understanding. How we do that exactly is not clear. Except for the lecture part, which is clear. So I will make a folder on Blackboard that says online lecture, and I'll date them. So the first one will be, I don't know what the Monday date is after break, but 23? Is that what you said? So the first one will be lecture, what's the month? March? March 23rd. Then there'll be a March 25th, and March whatever, 27th, and so on. Quizzes will still, in case you missed it, quizzes will still be in effect. I'll figure out how to deliver them. They'll be on Mondays, but not during spring break. Whether it's timed or not, I don't know, but I'll still deliver them so I can still split your grade up over a wider range of assessments. I think that's what people like. If you have questions for me about content, then you should email them to me and I'll be very responsive. Okay? So does that take care of that kind of stuff? The laboratory, I'll talk to you more about on Friday. But now we know what's happening with the lecture and the quizzes and the exams. Okay, good. So we were gonna take a quick look, and I'm sure I now have access. Let me take a quick look at a picture. So Blackboard, this is where you'll look for lecture content. You'll scroll down to wherever our class is, go to the Chase section. Right now there's lecture slides for Chase section. I'm imagining the online lectures will be contained within that folder. Seems like it's the most, that's where you're used to looking for stuff. So we'll click on there and we'll find it. What's that look like now? A, B, and C. So then I might add a content folder here that says online lectures, and then they'll be dated. So I think that is here. Laboratory protocols and results, so that must be where our gel pictures are. Here's the gel pictures. There's gel 1, gel 2, gel 5, and gel 6. So here's how I remember all those gels going down. Was I was stationed at one set of gels and Brendan at the other. In the morning at 8 a.m., the the gels that were destined for Kamasi staining were 1 over by me and number 2 over by Brendan. So if you're in group 1, 2, or 3, you came to me and loaded your samples on gel 1. If you were in group 4, 5, and 6, you went to Brendan and you loaded your samples on gel 2. You also loaded samples on 2 other gels, and they were numbered 3 and 4. They are not here. At least they shouldn't be, because those weren't stained. Those were the ones we're going to use for the Western transfer, and so they're being transferred right this minute. All the proteins on those gels are being transferred right this minute. And if you're at the 1050 group, the stations were the same. Group 1, 2, 3 came to me. 4, 5, 6 went to Brendan. I had gels the Kamasi was 5 with me, 6 with Brendan. So you should be either 5 or 6. If you're group 1, 2, 3, you're on 5. If you're group 4, 5, 6, you're on 6. And then the Western was on my side for gel 7. And then if you're group 4, 5, and 6, your gel for Western was gel number 8. Okay? That's how I remember it, and that's how they currently are. So all you have to do is remember who you were standing with. We'll just look at one of them. And it's smaller. So this is a fairly good representation of what we saw. Remember the blue, maybe not as clear. It looks better on my computer screen than it looks here. But what you can appreciate is that there's one lane with just about 11 bands in them. This one's actually yellow in color. That one's red in color. And we went through the protein standards and looked at the colors and what they meant. And hopefully, let me double check. Hopefully I loaded a picture of the gel marker, but it doesn't look like I have yet. So, it doesn't look like I've loaded a picture of the ladder by itself, indicating exactly the sizes of those bands. But I remember the red one's 80 kilodaltons, and I did write down in each section what those other bands were, including the identity of the yellow one, but I just don't recall anymore. But I will load the photograph that I placed on the front bench that you were all asked to come see. And so you'll have that. And then there's 6 lanes, so it was a group, and this was gel 1, so this is group 1's glucose and lactose, presumably. This is group 2's glucose and lactose, and group 3's glucose and lactose. And what you'll notice, especially when you see a nice quality image, is that there's lots of blue bands everywhere. I mean, there's a really big fat one here, and a really big fat one there. But there's a lot of other bands that are present in the lanes, and they become more clear again when you're not looking at it through a TV projection. Those are the proteins that are present in the bacteria. And you'll notice that there's largely the same proteins in all lanes. Like, here's a protein in the glucose lane, but so that's certainly the same protein in the lactose lane. Here's one in glucose and lactose. These two are clearly present. Those two are clearly present. And that's what we expected, that most proteins made in E. coli, regardless of what carbon source they were feeding on, would be the same. Both bacteria are growing, so they need things to grow. Both bacteria are making proteins. They need the proteins that are used in the ribosome, so those proteins must be present, and essentially all of the proteins. And our only question is, do we see a band in one lane that we don't see in the other? For example, our idea was that beta-galactosidase would be a protein that might be made in bacteria grown in lactose, but not glucose. And so if that was made to high levels, you might be able to see it on a gel that's stained with Kamasi. Kamasi stain is not the most sensitive of protein stains, so there's probably a lot of proteins that we can't see that are staining with blue, but below our level of detection. And to be able to begin to think about that, you've got to have some sense of how big beta-galactosidase is, and you've got to know what those protein markers are, right? So the point of running those protein standards is to get a sense of the mobility of different sized proteins in a gel matrix. So I think the biggest one was 245, and then there was a 190, maybe a 130. I don't know what they are, but if we knew all that information, we could look at this and speculate about whether or not we actually saw beta-galactosidase, and that's what you should be doing. These are the pictures you're going to use to make the figure for, the next assignment, which you will email to me. So I suggest you embed that figure into some sort of document, whether it's a Microsoft Word document or a PowerPoint figure, and send that to me either as a Word document, a PowerPoint, or a PDF. I can open all three of those, certainly. If you send it to me, a lot of people are really fancy. They send it to me like on a Google Doc. I don't even know how to do that, so don't do that to me. Because I'll just send it back to you and say, oh, now it's late because I can't open it. And I'm just not sophisticated enough. And I'm 60. I don't know if I want to become more sophisticated. So could you just send it to me in one of those formats that I've requested it in? And if you're not here, please do that too. Okay, so that's that. All other gels kind of look like this. So maybe surprise. I was kind of surprised because in years past, we'll often see unequal loading, but you can look at these lanes and say that people did a really good job of loading equal amounts of protein. I mean, even if you just looked at this band, you say, oh, that intensity of blue, probably representing a single protein, is pretty equivalent to this one. In fact, so is that one, and so looks like everything's pretty well equally loaded. Maybe this lane has more than this lane. This is the partner lane for that. You can actually see a streak at the top of the gel for this lane, which is typical of proteins that get overloaded. So that group just had such high concentrations of protein in their lysate that it began streaking. But even that one's really nice. And so if beta-galactosidase is there, I don't have any question that we'll see it, at least in the Western blot, where that technique is much more sensitive than the Kamasi. Okay, so we've looked at that. We've talked a little bit about how these are saved as JPEGs online, so you can move those into any document you want. Everybody should be able to manipulate JPEGs. Good luck, have fun with it. All right, that takes care of that. Doesn't look like I have the protein standards on there, so I'll remember to get those on post-haste. All right, so here we are. We're back actually talking about new material. I think we're closing in on finishing up with this protein structure part, and we're going to move into function part finally. But I've really pushed this idea over and over and over again of functional groups. We started with water, remember? Water had this polar bond, and that imparted some sort of character on water, gave partial charges to the hydrogens and the oxygen, which allowed that molecule to do something. It's not just floating around, having no interaction with anything else. In fact, it can interact with lots and lots of other things, and that becomes important. We transitioned into this idea that, boy, there's lots of polar bonds and amino acids too. We started putting amino acids together into polypeptides, and then we noticed this repeating functional pattern of carbonyl groups and amine groups that occur flanking every single peptide bond. I said, oh, that's interesting. There's something that we know has function, right? These carbonyls, the oxygen has a partial negative charge, and the Carbon has a partial positive charge. And with the amine groups, those hydrogens also have partial positive charge and can participate in hydrogen bonding. So that was kind of interesting. And in fact, if you, I'm going to segue out of that for just a second to say, when we thought about a peptidyl transfer reaction, remember it was the partial positive charge on the carbonyl group that attracted the electrons from the amine, the nitrogen, in the amino acid that was coming into the A site. Remember, there was an attack from the electrons of the nitrogen to the back side of that carbonyl. Well, they're attacking back there because that's where the positive charge is for the carbon, because it's involved in a polar bond with the oxygen. And so that's what sets up the chemistry that allows you to take polypeptides and stick them onto amino acids during the process of translation. So all these things that you're learning about really help you to begin to understand additional concepts. I guess that's the whole point. Making sure you understand the fundamentals of the structure of water and how that applies to everything else. Anyway, so these repeating carbonyls in the amine groups in the polypeptide, then we've transitioned from that, recognizing that they're a regular pattern, to then how does a amino acid stabilize structures, and one of them is by twisting on the backbone, forming an alpha helix, where those carbonyls and amine groups actually come within close proximity of one another such that they can form hydrogen bonds. And that stabilizes that polypeptide into this alpha helical structure. And then the very same groups are used to form these beta sheets, right? So it really is a cohesive story built out from a polar bond and what that really means. Okay, so we're wrapping that idea up, and I thought we'd wrap it up while I'm talking. You could see these structures as they fold on one another into this really complicated, three-dimensional, in this case, tertiary structure of a polypeptide that includes the alpha helices, some beta sheets, some unordered turns that are necessary. Once you get to the end of a polypeptide, you gotta come, turn, and go back in. And so all these kind of unstructured loops are also amino acid residues. They just aren't forming alpha helices and beta sheets, but they're absolutely required for a long stringed molecule to turn into some sort of a ball-like structure. This is what we mean by a globular structure. If you took this polypeptide and shook it out from here, that's going almost to Memorial Hall from here, I would guess. But once you twist it and fold it and turn it into a ball, it occupies a much smaller space. And it's that finished folded protein that acquires function, and that's where we're transitioning in, is how do these proteins have actual activity. The activity of this one, as we pointed out in the lecture earlier on, is to convert glucose 6-phosphate to fructose 6-phosphate. It's one simple activity. It breaks and forms a bond. That's all it does. And it only can work with glucose 6-phosphate. It has no other substrate, and it doesn't make any other product. So enzymes in general are one-show ponies. They do one thing, but they do it really, really well. They're built to do it. The portion of this molecule that actually does that is buried inside this cleft. So this part of the molecule never sees glucose 6-phosphate or fructose 6-phosphate. It all happens in here. So then what's the point of all these portions of the molecule? Well, it's really just to hold that active site in place and allow it to grab on to glucose 6-phosphate, make that chemical change, and then release the product. If you removed portions of the molecule, even that were outside of the active site, the protein wouldn't work anymore. It wouldn't adopt the structure that it's supposed to adopt so that it can, in fact, recognize its substrate. Okay, so to put it all kind of together, we talked about, what's happening with the polypeptide when it comes out of a ribosome. It's battling with its environment, which is water primarily, and itself, to try to find some sort of ordered structure that it could form that would allow it to remain stable in solution and float around the cell and find its targets and do its thing. And so this, what you're about to see, is an energy landscape, which is meant to show you that an unfolded polypeptide really has a high energy state. It's making all these battles with itself and with water, trying to find a more stable form where it can reduce the total energy of the system, which is not just the protein, but also its interaction with the environment. So I'll start the movie, but then I'll pause it in a bunch of places